1 second, sir. 1 second. Sir, this is Ginger Manilius. General
Okay.
characters. Yeah. Next,
Type study
Pelia.
name.
Sir?
Janganmanials, type study name. Plant name.
Plant name.
Sphagnum. Sphagnum.
Sphagnum. Only one.
No. Sphagnums and sphagnum.
Right. Sphagnum enough.
Yes.
So Janganmanials is the largest order of liverworts. So mainly Janganmanials only one type study, that is the sphagnum. Right? How look like the sphagnum? Is it dorsiventral and erect?
Erect, sir.
Erect. What about others? Rest of what we completed so far? The structure of the thallus look like. Just compare with sphagnum.
That is dorsal.
Dorsiventral.
Clearly I said the Markansia, Pellia, and Anthoceras, these three are not a erect nature. These are all dorsiventral and prostate. And clearly said from the ventral side, rhizoids, dorsal side, midrib and sex organs are developed. But this sphagnum is quite opposite. So before structure, what we study?
Habitat.
Distribution, habitat.
Habitat.
Right. Especially this sphagnum cosmopolitan distribution. In India, it is represented at about 20 species, especially in Himalayas. These species are aquatic or semi-aquatic, grows in mosses and swamps, ponds, lake, moist health, and wet hillsides. Let's see the external feature, external character. It is leafy gametophore. Leafy gametophore. So what about the Markansia, Pellia, and Anthoceras? Those are leafy. Can you see leaf-like structure?
No.
Those are mosses.
This is mosses and those are there is no leaf-like structure. Thallus only present.
Right. Just wait. So this is a sphagnum, right? Sphagnum external characters. Actually, before this sphagnum development, first actually the protonema stage is developed. Protonema is the temporary stage. Protonema developed is the temporary stage. This protonema developed from the spore. I will tell you later how the spores are formed. So spores developed into protonema, and this protonema developed into gametophore structure like this. Gametophore structure. Right?
Yes, sir.
So it is erect nature, aerial and erect, attached to substratum with multicellular rhizoids. We can observe here. Multicellular rhizoids with oblique septa. Right? So we can clearly in gametophore, we can observe clearly rhizoids already completed and stem and leaves. Stem and leaves. So if you see the diagram, rhizoids, stem, actually it is not a true stem, stem-like structure.
Yes,
Stem
sir.
and also leaf. First, let me complete about the stem. It is erect, and well branched. The branches are arranged laterally. Branches, well, branches laterally. Right. Here the two types of branches, divergent branches and drooping branches. The divergent branches, these branches grow out laterally from the stem and extended outward. Horizontal position, like this, horizontal position like this. These are divergent branches. And drooping branches, these branches grow out laterally from the stem and hang. Actually, these are hang down. These branches hang down. So these are called drooping branches. So anyway, how many type branches? Two, divergent horizontal growth and drooping hanging branches. And leaf, the leaves are arranged on the main stem and as well as branches. The overlap, these are arranged overlapped and arranged in spiral manner. And these are sessile. That means without stalk. So let me complete, let me finish with green color. So this is stem. Sorry. Green color leaf. Green color leaf. And these are arranged on branches also. This is hanging branch. Right. This leaf color, this green color parts are called leaves. These leaves are very thin, fleshy, broad base, midrib absent. To take the leaf midrib absent. There are no midrib. Midrib absent. And what is the function of leaf?
Photosynthesis.
Right, the photosynthetic function. So this is about leaf. So these three completed: rhizoids, underground pot, fixation, absorption of water, and aerial erect stem with two branches. What are the two branches? Hello.
Divergent branch and
Divergent branch and drooping branch.
Divergent branch and
And leaves are arranged on the branches and main stem. They are arranged as a spiral manner. Each leaf sessile means without stalk, without midrib,
without midrib.
and appear green in color because of consisting chlorophyll pigments. That's why it is assisted to the preparation of food material through the photosynthesis. So this is about external feature of the sphagnum, right?
Okay.
What is next?
Internal structure.
Internal structure, but here internal structure no need, not important one. And if any internal structure we will complete in the practical session.
Okay, sir.
So next one, reproduction.
Yes, sir.
What we learnt in reproduction?
Reproduction is by vegetative
And sexual method.
Right. What are the sex organs?
Antheridia and Archegonia. Ear,
Right. Right. Enough. And antheridium structure, you know, and archegonium structure also, you know. From the antheridium, male gamete, from archegonium, female gamete. And I already told you very beginning, the only formation different. Markantia different type of formation, sex organs, and Anthoceraus different type, and Pelia different type, and sphagnum also different type. Right. Let's see in sphagnum, first of all, the antheridium or the male sex organ developed on lateral branches. And lateral branches, lateral. For example, this is lateral branch. This is lateral branch. On lateral branch, antheridia develop. For rough diagram, this is from lateral branch, antheridia develop. And that branch called as male branch. And these branches develop near the apex of main shoot. These branches bear the leaves which are just like the foliage leaves, but comparison they are shorter. These leaves are brightly colored, commonly yellow or purple, brown, dark in color. In the axil of the each leaves, solitary, that means single antheridium develop. A mature antheridium consists stalk and body.
yes, sir.
Right? What are present inside the body? That is common.
Other cells.
Common. What are present inside the body?
Androgonial mother cells,
Right.
androcytes, and
Right.
anthrozoites.
Androcyte mother cells, androcyte and
Anthrozoites.
right. Next, after fertilization, what happened?
Formation of zygote will take
Before that, let me come. Before that, let you complete about the archegonium structure. Archegonium. Tell me about archegonia.
Archegonia is as usual flask shaped structure having 4 different parts: stalk, venter, neck, and cover cells. Central part, egg is developed.
Okay.
Then after the matured androzoid will enter into the open archegonia,
Right.
which is enters into the egg cell through the neck canal cell used to form for zygote.
Right. After that,
Zygote will develop into an embryo.
కొంచెం పని ఉంది వస్తావా అన్నం
please don't
పంపాలి
talk.
పచ్చి పులుసు కావన్నా
Zygote will
Continue.
develop into embryo. Then embryo is finally develops into a capsule.
Okay.
Capsule is having 2 parts only here, foot and smaller club-like globus foot is there, and swollen capsule is present. In the capsule, there is no seta here, and in the capsule.
No, don't explain about sporophyte. Okay.
Sir?
don't explain about sporophyte. First of all, so for Antheridium structure and Archegonia completed, right?
Yes, sir. Yes.
Antheridia, Archegonia completed, and fertilization also,
Fertilization.
right? Now,
Yes. Sporophyte. Instead of that, here sporogonium is there.
okay, right. So, are you able to see this diagram? Antheridia.
Yes, sir.
Okay, so this is on the stem. This is already told you. Just below the lateral branch. This is apical. This apical side. Lateral branches. Lateral branches. So antheridias are developed. This is antheridia, and these antheridia alternate with leaves,
Yes.
right?
Yes, sir.
Antheridias alternate with leaves.
Yes, sir.
So these leaves are any different with normal leaves?
These are very long leaves when compared to the normal.
Simple. These are branches, leaves, just like the foliage leaves. That means the gametophytic leaves, but in comparison, they are in shorter.
Yes, sir.
Okay,
Yes, sir.
and bright colored.
Bright in color.
Yellow, maybe purple, brown, dark green. But foliage leaves always green in color.
Okay.
Right? Each axil of leaf, only one antheridia developed. So this is leaf. Just wait. This is leaf. This is antheridia. One antheridia, just solitary. Leaf and antheridia. Leaf, antheridia. Hello, please. Right. Only single antheridia develop from the axil of the leaf. This is a leaf axil. One antheridia, and again leaf, axil, antheridia. And you know the clear structure of the antheridia? Antheridia, club-shaped stalk and club stalk and body is present. So inside this androgonial cell, inside body androgonial, androcyte, mother cell, androcytes, these androcytes develop into male gametes. Right, isn't it? Hello?
Yes, sir.
Yes, sir.
Right. The next antheridia, archegonia. Tell me again about archegonia.
Archegonia is flask
Flask
shaped.
shaped, right?
Stalk is having 4 different parts: stalk, venter, and neck, canal cells, and cover cells.
Right. 1 to 5 archegonia develop on tip of the short archegonial branch, tip of the branch. So not an axial side. Actually, antheridia developed on axial side, but archegonia developed on the tip of the branch. And the leaves of the archegonial branches are green and much larger than vegetative branch. That is a different. Leaf green in color and larger than vegetative branch. The leaves are called pericytium leaves. Pericytium leaves. These leaves protect the archegonium. So that is the function of pericytium. The archegonia which develop directly from the apical cell of the archegonial branch.
Excuse me, voice is not audible, sir.
Just wait 1 minute. Hello?
Sir,
Just wait.
okay, sir.
So here this protected leaves, just wait. Now tell me the parts of the archegonium. First of all, stalk, venter, stalk, venter, right. Neck, cover cells. And it is surrounded by protected leaves. These protected leaves called as
Pericardium.
right pericardial leaves. Protected leaves called as pericardial. And inside the venter, egg cell, venter canal cell, neck canal cells, and covered with neck cells. Covered with neck cells are present. Right. Then what happened before fertilization?
Cover
Zygote.
cell slits.
Right. At the time of fertilization, neck canal cells along with the venter canal cells disintegrate to form clear passage for the anthrozoids. They enter through the cover cells and reach up to the egg, but only one fuses. Of course, the number of anthrozoids or male gametes enter into the venter of archegonium. Only one fuse with the egg to form the zygote. Right?
Yes, sir.
Then form the zygote. Embryo developed. Embryo differentiated into sporophyte. Where is sporophyte now? The sporophyte on the gametophyte.
Yes, sir.
Right?
Okay.
Yes.
Now let's see the structure of sporophyte. Here the foot, capsule, mainly foot and capsules are there. pseudopodia, right? Right. First of all, food. Food is common to each and every sporophyte. Markantia also food, and Peli also, and Anthocyrus also. And what is the function of food?
Absorb, supply
Attachment
the nutrients.
nutrient
Right. Absorption and supply.
and attachment to the gametophyte.
Right. Just this one. Okay. Are you able to see this diagram?
Yes, sir.
Right. So first, let me complete the food structure. The food, it is bulbous-like structure. This portion food. Bulbous section, and it made up of parenchyma cells. Function absorption and fixation to the gametophyte. Next, immediately the capsule, of course, the small notches are present, that's not an important one, and this globose structure called as capsule. Capsule is spherical and dark brown in color, and it contains central columella. This portion, this center portion called as columella, this one, which is overarched by one dome-shaped spore sac. This is spore sac. Spore sac. What are present inside the spore sac? Dome-shaped spore sac. Spores. Haploid spore. This haploid spore developed from the sporogenous tissue. Spore mother cell. This spore mother cell undergoes to meiotic division. After that, spores are formed.
Yes,
Right?
sir. Yes.
Sir.
Next, the wall of the capsule is 4 to 6 layer thick. The outer layer of the capsule wall is called epidermis. The cells of the epidermis compactly arranged and contain chloroplast. Remember it. So this layers 4 to 6 layered. The wall is 4 to 6 layered. The outer layer called epidermis. Epidermis with chloroplast. So what is the function?
Photosynthesis.
Photosynthesis.
Right. For photosynthesis. That means the sporophyte can prepare food material like Anthocyrus. Anthocyrus also, after the epidermis, some of the are there.
Chlorophyll is there.
Right. Due to the composition of chlorophyll pigment, the Anthocyrus also can prepare their own food material.
Yes.
Sir.
As the same, the sphagnum also, as it consists chlorophylls, it also can prepare the food material like Anthocyrus.
Yes, sir.
Both
So
are
the sphagnum
independent.
also, what?
Both are independent.
But not completely.
Okay.
Partially independent.
Okay. Sir.
That means where it is fixed.
So gametophyte.
So that
On the
means it's
gametophyte.
dependent gametophyte, not
Yes.
separate. Dependent gametophyte only for water and minerals, but not food material.
Okay. Sir.
But in initial, the early gametophyte, early bryophytes, such as Markantia pellia, these are completely, that means for food material and also dependent gametophyte. They are completely dependent, but on the higher bryophytes incompletely dependent on the gametophyte. Right. Now dispersal, dehiscence of capsule next. After columella mature, columella dries up, breaks down and forms the large air cavity below the air sac. So this columella completely dry up and it's formed like a empty air sac. Air enters in this cavity through the rudimentary stomata. Rudimentary stomata means, actually already told you, epidermis with chlorophylls. And now and there the small pores also there. Small pores also present. So these are called rudimentary stomata. That means undeveloped, not well developed stomata. Through the undeveloped stomata, the air enter into the columella empty space. Now the walls of the capsule dries up under the influence of the sun, and spherical form of the capsule gradually becomes cylindrical. The imprisoned air in the capsule is compressed and held under considerable pressure due to the change in the shape of the capsule. The operculum also dries and shrinks. So this operculum present top of the capsule. This reason, this one operculum. Operculum. Right? The operculum also dries and shrinks. Due to this, the little difference in the tensions in this setup. That means inside the columella, the pressure increased.
sir.
Right?
Yes, sir.
Due to this pressure, these capsules rupture. The operculum is blown off with explosive force. Due to the force of the inner air pressure, the operculum fall down with pressure.
Close.
Now, as a result, the spores are released out. The spores are released into atmosphere as like a gun explosive machine. Because immediately, actually, inside the cavity, the air pressure, all of a sudden, the blast the operculum like a gun machine. So this type of called as a air gun mechanism. The spore dispersal mechanism called as air gun. Air gun mechanism. In air gun also, the pressure of due to the pressure of air, the bullet went out. Like the same here, the pressure of the air inside the columella, the operculum blown or fell down. Right?
Sir.
After that, the spores are released. Next, what happened? The spores developed into
Protoni.
right
Protonema.
protonema. The finally protonema developed into
Gametophyte.
gametophyte or not, gametophore. This better word.
Sir.
Gametophyte is the primitive. Gametophore is somewhat advanced. Developed gametophore. Right? Now draw life cycle. Please make it fast. Please sit back within 1 minute. Please put in group. Who is this? 595 9866269595.
Sir, name is sir.
Okay,
Okay,
right.
sir.
Sphagnum, right. Antheridial branch, archegonial branch, okay. Apical cell, primary androgonial, anthrozoids, mother cell. Where is anthrozoids? Okay. No, but did not write anthrozoids, androzoid, mother cells. Okay, not a problem. Zygote, embryo,
Sporogony.
sporangium,
Sporogony. Not sporophyte.
right. Sporophyte.
Okay, sir.
Sporophyte only capsule, right? Sperm, mother cells. Epidermis, spore sac, okay, right. Capsule, epidermis. And you have to write the this capsule wall instead of epidermis. 4 to 6 layer, the outer one epidermis, okay, right. And spore sac, okay, right. Center columella, right. The better when compared with the previous life cycle, this is the better one. Haplodiploidic, heteromorphic. Mentioned here the heteromorphic.
Okay.
Heteromorphic, haplodiploidic life cycle, right.
Trauma. Okay, sir.
Next. Alt of female gametophyte, male gametophyte, antheridium, archegonium, egg. Don't copy book. Already told you.
Sir, nothing is showing on the screen.
What? Somewhere here exactly copy the book matter.
Sir, the screen is totally black. Nothing is showing us.
Can you show on screen? No, sir. Excuse me, sir. Can you show on the screen? No. Hello?
Tell me.
Hello. Can you show on screen? No, sir. Life cycle once again. Please.
Sir, the screen is totally black.
You yourself draw the life cycle. Did not hear the class?
No, sir. Just I
Don't you
attend.
know how to draw the life cycle?
No, sir. Just I attend the class now itself.
I will send later. First of all, try it. What you learned so far about the sphagnum? Write the same matter in orderly as a cycle. That would be life cycle. Understand?
Okay, sir.
In orderly, first what you learned? The sphagnum external character. Right?
Yes.
Next. After external character.
Reproduction.
What have you learned? Reproduction, right? Reproduction, then
Development of Antheridium and Archegonium.
right. That only write on paper. First write the sphagnum plant. From the sphagnum, the sex organs are developed. What are the sex organs, you know? And what are formed inside the sex organ, you know? And what is fertilization? Fertilization process also, you know? And as a result of fertilization, what are formed also, you know? Then how they developed into next part. And again, how the plant. Gametophore plant, sphagnum gametophore plant. That is the life cycle. You start the class with the sphagnum gametophore and end with sphagnum gametophore. That is the life cycle. Now write. Right. Okay, I will send the life cycle end of the class. And after that, you correct it yourself.
Okay, sir.
Right. And what next? Gametophytic evolution and sporophytic evolution, right?
Yes.
Yes, sir.
Right. Hello? Sphagnum completed.
Yes, sir.
Right. What is the next rate?
Evolution of gametophyte.
Evolution of gametophyte. Do you know all gametophytic structure?
Yes, sir.
Tell me. Why are you laughing at? I am asking you the gametophyte structure.
Given,
In
sir.
Bryophyta, the gametophyte is green in color, thallus-like structure, dichotomously branched, dorsiventral, are like the root and the stem leaf-like structures, but it is not true leaf, true leaf or true stem.
Right.
Gametophyte is having both sex organs, antheridia and archegonia, and also having some vegetative structures like gemma cup. Gametophyte is having chloroplast. It is green in color. Because of having chloroplast, performs photosynthesis and can prepare their own food material. Rhizoids are also be present for the encouragement and also the attachment of any substrate.
First of all, so far we completed in orderly. First Markansia, Pellia, Anthocyrus,
Sphagnum.
Sphagnum. Already these four types studies we know. Before that, the simplest gametophyte is simplest gametophyte is Rixia. Rixia. This is very simple form of the gametophyte structure. And from the Rixias, Carcinia, Spirocarpus, some other also there, but no need to mention all one. Rixia. Right about just mentioned Rixia gametophyte. Rixia gametophyte also look like a Markansia, dorsiventral and dichotomously. Just when compared with the Markansia, this is small simple structure. That's it, because no midrib like that. And next one Markansia, and next somewhat complex Pellia, and somewhat complex Anthocyrus because it is much lobed. And next sphagnum. Sphagnum, polytrichum. Polytrichum. Hello.
Yes, sir.
Please away from sounds. Right. Was Rixia, Markantia,
Pilia.
Anthocyrus,
Pilia and
Pelia, Anthocyrus,
sphagnum,
Sphagnum,
polytrichum.
Sphagnum, Polytrichum, and Funaria. These are advanced gametophyte. Advanced gametophyte because this Sphagnum, Polytrichum, Funaria, aerial, not dorsiventral. Not dorsiventral. So this much only you have to write in examination. In examination, if they ask about the evolution of gametophyte, just write about the what you learned so far, Markantia, you know, and Pelia, no, Anthocyrus, Sphagnum. And just add extra about the Rixia. The Rixia similar character to Markantia. Not much difference. And since Sphagnum, like Polytrichum and Funaria, these are all same category. So just write the 1 or 2 points about the gametophyte and draw the diagram. Each and every diagram, Rixia diagram, Markantia diagram, Pelia diagram, Anthocyrus, Sphagnum. That means is the simple in evolution. In evolution, this way. The gametophyte developed from the dorsiventral. For example, this is dorsiventral. The simple one. The simple one, Rixia, dichotomous. Dichotomous. Next, much branched or much lobed. Next, slightly developed into erect nature. Slightly developed erect nature. So this is the evolution. Dorsiventral, simple, complex, somewhat more complex, and this is not a dorsiventral, erect nature.
Aerial.
So this is the evolution of gametophyte. The simple.
Sir, sphagnum, polytrichum, funaria, aerial plants, no, sir?
Yes.
Aerial
That
plants.
means erect and aerial side. Of course, these first Rixia, they also aerial, but they are in dorsiventral, prostate nature. The first primitive bryophytes are prostate, not erect. Sphagnum, Polytrichum, Funaria, they are the erect. That is the different. Erect
They
means
have
look like a
rhizoids or roots. Rhizoids.
What?
Sphagnum, polytrichum, funaria have roots or rhizoids.
All are rhizoids. There are no roots in Bryophyta.
Okay, sorry, sir.
All are rhizoids. What is the difference between rhizoids and roots? Of course, the same function and appearance are same.
Not as strong as like roots.
Not strong as roots, and also
Like structures not penetrated deep into the soil.
okay, and
Not penetrated deep into the soil.
and what here the small structural change. Of course, externally appearance same, but internally structural changes. Function same, but structure different. That means actually Bryophyta rhizoids without steel. But root with steel. That means with vascular tissue.
Okay.
That is the different. Right?
Okay.
So this is gametophyte evolution. Next, what is next? Next read. Any doubt in gametophyte evolution?
No, sir.
Evolution theories are there in reader.
What?
Evolutionary theories, reduction theory,
That
condensation.
means the simple reduction theory and what is another one?
Condensation theory.
Condensation theory.
Read about reduction. What first of all, what is reduction?
Reduce.
Reduce.
Yes.
That means what we say, first of all, this is what you see in the screen. This is the evolution of gametophyte from simple to complex.
Yeah.
If the evolution reversed, that means complex to simple, that is reduction.
Reduction theory.
Clear?
Okay.
One is forward, another one is backward. Simple. One is Rixia to Funaria, another one is Funaria to Rixia. Okay?
Okay, sir.
Right. And
Progressive evolution theory.
Progressive
progressive
evolution.
means simple to complex. That means simple structure Rixia to complex structure Funaria.
Funaria.
So
Right.
reduction theory and condensation theory are same.
No.
Okay.
The progressive theory means from simple to complex.
Right.
It means
Right.
condensation theory.
Okay. Is it clear or not?
Yes.
Right. What is next in it?
Evolution of sporophyte.
Evolution of sporophyte. Do you know sporophyte structure? Sporophyte structure.
Yes, sir.
Right. You know Markantia sporophyte and Pelia, Anthocyrus and Sphagnum, right?
Yes, sir.
Just add some more sporophyte like Rixia, Spirocarpus, Carcinia, Polytrichum, and Funaria. Just you have to learn extra 4.
Rixia, Sphagnum, Polytrichum, Funaria.
Right. Just wait.
Yeah.
Page number 211, there is evaluation of sporophyte. That drawing is enough, sir.
Enough. What they given?
Rixia,
Rixia.
Spirocarpus, Pteridophyta, Hypophilia,
Targionia and
Markantia pilia.
and Pelia. 5 are there, sir.
okay. Just okay. Just enough. First of all, tell me about Rixia. Just open book. How is Rixia sporophyte? That means parts of the Rixia sporophyte.
Epidermis is present, single layer epidermis
very
socket
round,
present.
disintegrated jacket layer of the sporangium.
First of all, parts. What are the parts of the Rixia sporophyte? Is it clearly differentiated into food sita capsule?
No, sir.
No, only capsule there, I think, right?
Yes, sir.
Yes.
So that is the first diagram. Only spherical.
Integrated jacket layer of the sporogonium.
Just covered with jacket layer, no foot and sita, right?
Yes, sir.
So this is simple. This is simple sporophyte. The first formed sporophyte is the simple. And the rest of the common. What are present inside this capsule part?
Spores are present.
Spore tetrads.
Right. Spores.
Tetrad spores.
Only spore
Spores are
tetrads.
this yellow color are spores. Then what is
And
given next
outer
one?
layer of calyptra. Outer
Outer
layer of calyptra.
Okay. So this outer layer covered with calyptra. Okay.
That's it, sir. Only 3 labels are
So
there.
blue color calyptra. And next, what is the next? This is Rixia. Okay, completed.
Spirocarpus.
Spirocarpus. Now read about Spirocarpus.
Spirocarpus is little
Food
well.
Seta capsule is there. Food
Food
Seta
Seta capsule.
Yes.
Okay.
Food Seta.
Is there Carcinia?
No.
Sardinia is there, sir.
Carcinia. Corsia. Carcinia given?
No,
No,
sir.
sir. Just to
directly
food Seta.
Spirocarpus.
Seta.
Seta food rocket.
Okay,
It
right.
consists of
Spirocarpus
capsules.
clearly.
Middle layer jacket is there and tetrads are there along with elaters.
Right. Spirocarpus food. Small
Seta.
Seta. Remember.
Okay, sir.
And
Sterile
capsule.
nut. Sterile nut cell.
Same tetrads are there. 4 tetrads.
Okay.
Another
Four
one
tetras
is
there and
Elaters are,
sterile nut
elaters
cell.
are there.
elaters also present. So red color elaters. Next, after Spirocarpus, next one.
Sardinia.
Torgenia.
Targoninia.
All right, about tell me about Torgenia.
Sardinia also consists of food seed.
A large
Excuse me.
Foot and
Capsule.
Seta and
Elector.
tetrad.
Consists
right.
of a jacket.
Sir, excuse me, sir. It is second one is the Targonia hypophylla. Second one you drawn now that is Targonia hypophylla and now you are going to draw Spirocarpus. Spirocarpus is it is spherical
There
and
is
small
no
theta is
elector
there.
in Spirocarpus.
Excuse me, sir.
Tell me.
Second drawing is Targonia hypophylla. Now you draw now this.
What is first Rixia, right?
Yes.
First Rixia,
After Rixia, what is next one? Spirocarpus, right?
Spirocarpus.
Spirocarpus, the Seta.
Sir, but Spirocarpus don't have any electors.
Okay.
Oh, that's why I'm saying.
Okay. Someone told elaters are there.
Yes.
That is confusing picture. That is A, B, C like that is there.
No,
They are
that's why I'm asking you. Just read
Targonia
the matter.
consists of
Don't see the diagram.
Yes, sir.
Hello?
Yes, sir. Targonia consists of electors.
Targonia hypophylla, sir.
Right. Next one after
Small
Spirocarpus.
Small theta is there. Foot is there.
Right. This small
Tetrad.
Seta, foot and capsule. That's enough.
Sterile nut cells
And
are there.
sterile nut cell.
Sterile nut cells are there. Nuts cells.
Okay.
Now T1 and that is third one is Targonia hypophylla.
hypophylla.
Tetrads and
They
elaters.
have electors. They
The
have
same diagram, Targionia. Remember
exactly
the same
same,
diagram.
exactly
Hello.
same. But there is
Let me
presence of
complete.
electors.
Okay, right. Let me complete first. When compared with Spirocarpus, Targionia, same foot, Seta, capsule.
Yes, sir.
But the somewhat elongated Seta. Somewhat
Yes, sir.
elongated Seta. And inside the capsule, elaters are developed.
Yes, sir.
Right? Any pericytium layer around the capsule?
There is no layer. Only
Right.
jacket is present.
Right. No protection layer. And next,
Yes, sir.
after Targionia, Markansia, right?
Markansia, polymer.
Already Markansia, we completed Markansia.
Yes, sir.
Same foot and same elongated stalk.
Yes, sir.
That means Seta and capsule with elaters and
Electors and
capsule
spores.
wall. Calyptra and pericytial leaves around protected by.
Capsule.
Right. Next, what is next after the Markansia?
Cilia.
Pelli also completed.
Yes, sir. They have
But
also
what is the difference
cilia
between
foot.
Markansia and Pellia?
Meristematic tissue is present.
No.
Sir,
I'm
does
asking about Pellia, not
the
Arthoceraus.
cilia consist of electors?
Elatophores are there.
Right.
Group of elaters are there.
Right. Elatophores. And another difference with Markantia.
Sporogenous tissue. Columella.
Double
No
layer.
columella in Pelia.
Double layer cell.
Right. Jacket layer, double layered.
Yes,
But in
sir.
Markantia, single layered. Now tell me,
sir.
the evolution is simple to complex or complex to simple? Simple to complex. That means while the evolution gradually increased, the sterile cells number also gradually increased.
Yes, sir.
Sterile cells. That means first of all in Markantia only single layered. Next double layered. Next, what about the Anthoceras? Next,
Anthocer also is double celled embryo converted into 8-cell embryo.
I am asking Anthoceras sporophyte.
Acha.
Anthoceras sporophyte is having food, but no seta is present.
Just right. Seta absent. And advanced character is the seta replaced by sporophyte. That means meristematic tissues.
It's
Sporogenous tissue and
chlorogenous
chlorogenous
tissue are present beneath the
Right.
There is operculum. There is operculum.
Columella.
Simple question to you. When compared with Pelia Markantia, what is the complex structure in Anthoceras? What is the complex structure in Anthoceras? Hello, please.
గుర్తు పెట్టుకొని ఉంటది మాట ఆలోచన నేర్చుతుంది ఆమెను బుక్ చేద్దామా చేద్దామా అనే ఆలోచన
Padmavati, do you have manners? Hello?
Yes, sir.
Yes, sir.
Please be silent.
Yes, sir. Sir, in Anthoceras sporophyte of Anthoceras, Anthoceras, there is an operculum. There is the presence of operculum
Apophysis.
and apophysis.
Peristome is there.
Anthoceras chlorogenous tissue is present and sporogenous tissue is present around the columella, sir.
In Anthoceras, operculum there?
Yes, sir.
No operculum is there.
Operculum is there.
Anthocera operculum is there and peristome is also there.
No, you are completely wrong.
Operculum is not there.
Anthoceras is there, sir. Operculum is there in reader.
Operculum is there for funaria.
Anthoceras.
For Anthoceras, it is not there.
Already Anthoceras type study
Excuse me,
completed.
sir. Funaria.
That's why I am saying it.
Oh, sorry. Funaria operculum is there and
Right.
Anthoceras there is no operculum.
Right.
In
So
reader they
first
are
of all,
given.
let complete the Anthoceras. Anthoceras, just the one. I am asking the simple question to you. What is the complex tissue? What is the sterile tissue in Anthoceras?
Parenchymatous chlorogenous tissue.
Right. Parenchymatous chlorogenous tissue. That this chlorogenous tissue absent in the previous sporophyte, right?
Sporophytes, yes, sir.
And next one.
Sporogenous tissue and also present of columella.
Right, columella. And meristematic tissue.
Yes, sir.
Right.
Yes, sir.
Next one. After Anthoceras,
Funaria.
There is funaria.
Funaria. Now tell me.
Small
Is,
beta is there. Long capsule is there.
Sphagnum not there.
Sphagnum not given.
up to funaria only, sir.
Anthoceras and funaria only.
Actually, before that, we have to learn about sphagnum because already you know the sphagnum structure diagram. So after sphagnum, polytrichum and funaria. Only one enough. Either polytrichum or funaria enough.
yes, sir.
Now compare with sphagnum or Anthoceras. What is the extra complex sterile tissues in funaria? Just see the book and tell me.
Operculum present.
Right, apophysis, extra part.
Operculum.
Operculum, extra operculum, of course, sphagnum also operculum. Right, operculum also, and
Columella.
Archaeosporium.
right, columella,
Archaeosperm and air sacs are there.
Trabeculae.
trabeculae. That's one.
Yes.
Trabeculae.
Air space is there in Funaria.
So these are the extra part, extra complex structure of this sporophyte, right?
Yes, sir.
Yes,
Yes.
sir.
So
Yes, sir.
what happened? Gradually, the sterile tissue number increased.
Yes, sir.
So this is progressive theory. And remember the scientist who discovered this theory? I think Campbell, Bohr,
Tapasya.
Kashyap, right?
Yes.
And the same another theory, regressive. Regressive means reverse. Remember, one is progressive from the simple to complex. Regressive theory that means different, completely reversed. According to progress, the simple one Rixia, advanced one sphagnum, Funaria.
Funerary.
But reversed mechanism, regressive theory, simple one
Funaria.
Funaria, advanced one
And
Rixia.
Rixia is the
Right, advanced one Rixia. And what you observed so far in sporophyte evolution?
in sporophyte evolution from simple
Actually,
to
most of the scientists accepted progressive theory, rejected regressive. According to progressive theory, what you observed in terms
simple.
of sporophyte?
Sporophyte is
Sporophyte are
simple.
developed simple to complex. Some parts are evolved during first Rixia to Funaria.
Right, some parts are evolved. What are those parts evolved?
Like
Chlorogen.
columella,
Right. Mainly,
meristematic tissue.
right, meristematic tissue and chlorogenous tissue.
Okay.
Chlorophylls, chlorogenous tissue. So, chlorogenous tissue mean
Photosynthetic.
photosynthesis.
And form photosynthesis.
Right. Actually, in initial stage, Rixia to Pelia depend on gametophyte for food material and water.
Yes,
From
sir.
the Anthoceras to Funaria,
Yeah.
they can prepare their own food material, only depend for the water.
Attached water.
Right. In plant evolution, what is next? In plant evolution, we completed so far algal member complete and Bryophyta complete. And next,
Pteridophyta.
Pteridophyta. Now tell me what is pteridophyte?
First vascular cryptograms.
First to vascular plants,
Plant body is
Vascular cryptograms.
well
Right.
differentiated into root, stem, and leaf.
root, stem and leaf, clearly differentiated root and stem and leaf.
Sporophyte is independent.
Spore, that means main plant body is sporophyte, that is independent.
Yes, sir.
And
What
sporophyte
about
is
Bryophyta sporophyte?
That is
Bryophyta
dependent.
sporophyte, dependent. Here, remember, here just observe here the first initially primitive sporophyte completely dependent, advanced sporophyte partially dependent. When reached Pteridophyta, this sporophyte completely detached from the gametophyte and can develop independently and develop into pteridophytes. This is evolution. That means when reached the Pteridophyta, the foot developed into root system, seta developed into stem, capsule developed into leaf and flowers. Right?
Yes, sir.
So Bryophyta sporophyte, when reached the Pteridophyta, clearly differentiated
Depend
into
upon gametophyte.
root and shoot and leaf system. Okay? Is it clear? Any doubt?
Most of the pteridophytes are terrestrial in nature.
Yes, terrestrial. Of course, very few are aquatic also. That another matter. That mostly terrestrial, right? Next.
Herbaceous, except a few woody trees. All the living pteridophytes are herbaceous, nearly
Okay.
herbaceous. And mature sporophytes are independent, though in early stage of development they are partially or completely dependent on gametophyte. The stem are dichotomously or laterally branched.
No explanation. Now no explanation. But now, first of all, are clear about sporophyte evolution in Bryophyta. That is first one.
Yes, sir.
Second one, are clear about how the sporophyta bryophyte developed into Pteridophyta sporophyte.
Sir, Sporophyta foot is developed into the root.
Yes.
Phyta is developed into the stem.
Okay.
Developed into leaves, flowers, and inflorescence.
Right. That's it. Now next black is Pteridophyte, right?
Yes, sir.
Pteridophyte definition also clear.
Yes, sir.
Right. Now the Pteridophyta general character before the distribution. Distribution, this is a common, nothing to explain about distribution. Any type of distribution, you can read yourself. Don't waste your time under my time. Just in next one, habit, that means external character also, root system, how is root, you know, how is stem, you know, how is the leaves arrangement also know. Next one, the reproduction. Sporophyte, what is the reproductive part?
Sporangia. Sporangia develops either on
Right.
ventral
Sporophyte,
side space of the axis of leaves.
sporangium developed this total part called as cone and strobile. This is important. Cone or strobile. In Pteridophyta, already definition clear, right? Individually, root system, always root, shoot, leaf, and finally matured cones or strobile. This is the reproductive part, like a flower. The cone developed by special leaves, these are called sporophylls. Spill means
Is also called fronds, no sir?
what?
Fronds, F-R-O-N-D-S.
Right. They are the sporophyte leaf. Fronds are the sporophyte leaf. Sporophyll is the reproductive leaves. Totally
Yes.
different.
Yes, yes.
Okay, this is the this way. Green color box only sporophyte structures, and red color reproductive parts. Cone made up of sporophyll. Phyll means
Leaves.
right. That's it. Spora means sporangia. Sporangium.
Sporangium have sori.
Right. Sporangium plus leaf
Sporophyll.
called sporophyll. What are present inside sporangium?
Sori, the group of spores are present.
Spores.
Sorus.
First of all, what is the ploidy of sporophyte?
Diploid in nature.
Right. Diploid.
Right.
Then after formation spore, mainly how the spores are formed? By mitotic or meiotic?
Mitotic leaf.
Meiotic cell.
Only spores are developed by meiotic division.
Meiotic cell.
Meiotic, that's why the spores become haploid.
Haploid.
Right?
Yes, sir.
Now this haploid spore further developed into?
Gametophyte.
That's it.
Yes, sir.
Gametophyte. The ploidy of gametophyte, haploid. Now gametophyte dependent or independent? Independent. That is the important feature of the pteridophyte. In Pteridophyta, sporophyte, gametophyte, both are independent. Right?
Yes, sir.
Now read about the gametophyte This gametophyte look like a Bryophyta. Right? What are formed on the gametophyte?
Stem, leaf.
No, it is not differentiated stem, leaf. What about Bryophyta? Bryophyta is there any stem and leaf and root?
Prostate, erect,
Right. Same like
Versiventral,
Bryophyta,
Bryophyta.
gametophyte. The Pteridophyte also same, similar to Bryophyta. When reach maturity, what are formed on the gametophyte?
Sporophyte.
Just remember.
Branches and leaves are formed. Instead of
First of all, recall Bryophyta, gametophyte.
sir.
After external structure, what you read?
Reproduction.
Reproduction. Here also same. After formation gametophyte, reproductive parts are developed. What are the reproductive parts?
Acridia.
Same Athridia, Archegonia. Same structure. Same club shape, same flask shape, same neck cell, neck canal cell, venter cell, egg cell. Antheridium produces male gamete. Next,
Fertilization.
fertilization means male and female fused. After that,
Zygote formation.
zygote developed into
Embryo.
Embryos.
embryo, further developed into.
Sporophyte.
Now this Pteridophyta class start from
Caps.
Gametophyte.
start from
Force.
Pteridophyte.
Pteridophyta class started from gametophytic stage or sporophyte stage.
Sporophytic stage.
Sporophytic stage and end with
Gametophyte.
Sorry. Sporophytic stage again. End with sporophytic stage.
end with sporophytic stage. Now the cycle clear.
Yes, sir.
Okay, don't worry, I will explain next class. Okay,
Okay, sir.
right.
Thank you, sir.
Sir, life cycle, sir.
Thank you, sir.
Life cycle of sphagnum. Thank you, sir.
Sir is not there.
Hello, madam.